Hi, today is the fifth medical talk. It will be in the second video on the eye, focusing on the retina. The retina is a light-sensitive portion of the eye. So this is the retina, this whole thing in yellow, the inside covering the inside of the eye is retina. So it contains cones for color vision, rods are for black and white vision, and when the cones or rods are excited, the signals will be transmitted to the brain. And the pigment melanin covers the whole retina to prevent reflection of light. So albinos are those people who cannot synthesize the pigment melanin. So their eyes also don't have melanin inside in the retina, covering the retina. So their vision is seldom better than 20/100 to 20/200. The retina is composed of 10 layers. So this at the bottom is the direction of the light. It will hit the ganglion cells first. Finally, it hits the cones and the rods at the back. So there are 10 layers. This is the retina. The foveal region of retina is 1 square millimeter in area. And then there's a center of it which is only 0.3 millimeter in diameter. The cones in the fovea is the center of the retina, have long and slender bodies so they can detect details. Whereas the cones in the periphery at the side are fatter. Okay, you can see the picture on the right. The cones at the bottom are very thin. In the middle are very thin. And comes to the right hand side is become very fat. And the ganglion cells open up a space in the center so that the light can pass through. So at the center is very good vision. Okay, this is this picture on the left. You can see where the fovea is. It's at the center. So yeah, the blood vessels and the ganglion cells are this place. So what are rods and cones? Rods and cones, they are actually photoreceptors. In the rods, there is only one photochemical. In the cones, there are 3 photochemicals because it can detect 3 colors. And there are as many as 1000 discs. So this is a picture of a cone. On the left hand side, there's an outer segment. This outer segment has discs. These discs, they can sometimes be up to 1000 discs. In the middle inner segment, we have the mitochondria, then the nucleus, and the synaptic body. So 4 parts. The outer segment is the photochemical segment. The inner segment is the mitochondria, where they generate energy. The nucleus doesn't undergo any cell division. So when the cones die, it cannot regenerate again new ones. And the synaptic body is the connection to the subsequent neurons. So what is retinal detachment? Sometimes the retina can detach from the skin, right? Can detach from the layer from the eyeball. Okay, the outer cones means the cones get blood from the arteries, nutrients. So what can cause retinal detachment is 2 things. One is injury to the eyeball that allows fluid or blood to collect between the neurons and the retina. So when it collects in between, then the thing expands and then it gets detached. Occasionally, it's because of low pressure in the vitreous humor, and this pulls the areas of the retina toward the interior of the globe. So detached retina can still survive for a few days and until surgery is done. But after that, after a few days, the retina will start to die and even surgery cannot help. So how does the photochemical reaction works? So light energy causes the photochemical to decompose into a different molecule. So as it decomposes, it excites the cones and the rods. It changes into the photochemical will change into another isotope, and then the changed isotope will be converted back to the photochemical using energy and enzymes. So vitamin A is an intermediate step. If you see here, see light energy comes to Bartholin. It changed to so many things, come down to vitamin A. One of the intermediary products is vitamin A, and vitamin A changed to retinol, and then retinol changed back. Okay, so vitamin A is a interconversion from photochemical to or from vitamin A. Lack of vitamin A can cause night blindness, but we don't have to worry so much about vitamin A because the liver normally stores enough for a few months. So color vision, how do we have color vision is because there are 3 color pigments in the cones. Every cone got one color. So, and the colors are dispersed at a constant ratio for most people. So the wavelength, each color have a peak wavelength of 445, 535 and 7579 nanometers. These are also the peak light sensitivity for each type of cone. So if you have, for example, 580 nanometers, it will stimulate the 3 colors, 99%, 42% and 0%. When you stimulate this ratio, the brain interprets it as orange. Some people don't have some color cones, so they can't see those colors. If you don't have green color cones, then you can't see green. So these are the wavelengths, right? So light and dark. So our eyes can also adjust for light and dark adaptations. So in a bright room, all retina is a photochemical, will be converted to vitamin A. In a dark room, vitamin A is converted to retinal. So when you leave a dark room like a cinema, you find that everything seems bright, even the darkest spot. But you are able to adjust within a few seconds because the photochemical starts to change back. And then the eye is very marvelous because the eye can see in very bright sunlight and it can also see in very dark starlight. So ganglion cells, where's ganglion cells? Ganglion cells are the first cells. Okay, coming from the direction of the light, they are the first ones. What are they? Okay, actually what do they do They help detect fast changes in light intensity. For example, if a fly across your face, immediately you notice it. But a fly sitting quietly, you don't notice it. Right? This is because of the ganglion cells. And it also helps the cones to differentiate colors. It can be excited by certain colors. Conclusion. What have we learned? First, we learned the retina. What is the retina? It's a light-sensitive region covered by the melanin. There are 10 layers. We learned the foral region. There are many cones, especially in the center. We learned what are rods and cones. They contain photochemicals. We learned about retinal detachment, which can be caused by 2 things, injury to eyeball or VH pull it inwards. We learned about the photochemistry of vision, how the photochemical decompose, and vitamin A is an intermediary. We learn about color vision, how the ratio wavelengths excite 3 types of cones. We learn about light and dark adaptations. Photochemicals is because of photochemical not in the right state. And we learn ganglion cells. They help in detecting light sensitivity and color. So the last point, we should see that this is a very complicated organ and appreciate what we have. Thank you.